All right. Hi, a big warm welcome to everyone today. My name is Jen Barnes. I'm the director of the Burnet Diagnostics Initiative. I joined the Institute about 2 years ago to focus on the translation of our research discoveries. And I've been working in that field for about 20 years now, in general, focusing on women's health. So I'm really honored to join both the Institute and you today to celebrate and highlight some of the amazing work being done. I'd like to begin by acknowledging the traditional custodians of the land from which we all join today. I would also like to pay my respects to elders past and present. A very warm welcome to all our donors and supporters joining us today, and I'd like to do a few special shout outs. First, welcome to Mary Padbury, the chair of the Burnet Institute. I'd also like to welcome Alison Larson and Ben Foskett joining us from our Burnet board. Not sure if she's online yet, but I'd like to welcome Professor Susan Crowe, AO. She retired from the Burnet in 2018 after a stellar career of research, and many will have met her over the years. And importantly, I wanted to highlight she spoke at our very first International Women's Day donor event in 2013. So this is actually the 11th event. I'd also like to welcome Dame Carol Kiddu, the patron-in-chief of the Burnet's Healthy Mothers, Healthy Babies program. Earlier this year, Dame Carol was appointed an officer in the General Division for her service to human rights, community development, and international relations. Also, I need to do a bit of housekeeping. So we're going to hear from a number of speakers today, and we'll introduce them as we go. There will be time for questions, but feel free to also pop them in the chat as we go, and I can read them out at the end if we have time. Also, please remember to mute yourselves during the talks. And I also understand some of you may need to leave early, and that's totally fine. Ash will be following up after the event today with a recording of the session, as well as some further links. So it's my pleasure to first introduce and welcome our CEO and director, Professor Brendan Crabb. Really pleased you could join us today and help launch the event with the international theme this year of embracing equity. Brendan? Sorry, is it Brendan? We're just having problems hearing you. You maybe turn off your video and we'll see if your sound comes through better.
How is that? Is that better?
Perfect.
All right. I don't know what's going on there, but I did the IT crowd thing. I turned it off and turned it on again, and it's now working. Yeah, pay my respects to elders past and present of the Wurundjeri people of the Kulin Nation, and you know, not the topic for today, but very heightened area of importance for us at the moment as we enter a pivotal moment in Australian history around the voice. Our theme today is equity, and of course, the challenge for more equitable Australia is a huge one that is both based on gender and recognition of our First Nations people. It is incredibly special for me to be here. I passed 15 years as the director of the Burnet a week ago, and I've never spoken at this event before, not because I haven't wanted to. I've always wanted to, of course, but it just hasn't been appropriate. The nature of the events had a magic to it that I've really supported, and you know, without trying to define what that is, you know, it's had a lot to do with the scale of the challenge, the scale of the mountain that needed to be climbed. But we have reached that point. You know, the team at the Institute's reached that point, not me. I'm saying, you know, it's time for us all to be involved very visibly, and that's why I'm here today. Please don't think that in previous years it hasn't been top priority for me. It absolutely has. But to sort of steal a phrase from Caroline, you know, try very hard to lead from beside or even behind in relation to to this and many things, actually, the more I learn from others. So very special for me to be here. You know, equity is in our DNA. It's in our vision statement, of course. It used to be equity through better health. It's now a more equitable world through better health. And the nature of the programs that we run and the funding we get is often directly related to it. So related to equity across the board, but especially gender-related issues. And in fact, even if it's not related to the project we're actually doing, it's a part of the accreditation to get the funding in the first place. So for a long time, I've had a degree of comfort that we as an organisation take these issues very seriously. We sort of ahead of the curve, if you like. But actually, internally as an organisation, that turned out not really to be true. When you looked over the last decade or so at our figures for women in leadership, for pay differentials, all the sorts of hard markers, we were sort of no better than anybody else, really. And that came as, whether a shock's the right word, but it was a balloon being pricked. I can't remember how long ago it was, but it was really obvious that we were no better internally, really, even though we had this great desire to be, and even sense that we were somehow ahead of the curve because of the nature of the work we did. So we took a deep breath and decided we did need to do all we could internally to do what we could to make a better organisation. And so that's what we did. We started by forming, I don't know if we started this way, but we eventually formed a Gender Equity, Diversity and Inclusion Committee that is chaired by Caroline, and has had, and I sit on that committee, and it's sort of overseeing, a lot of places have one, so it's just in the sense of committee, but actually it's been a very powerful force overseeing the sort of changes that we had. I must say the changes that we've instigated have also come from our board that's taken this very seriously. So it's not just management, and our board is close to parity. All our leadership positions at the Institute now, almost all of them are close to parity. Noting that our workplace is actually predominantly female, 66+% of Burnet staff are female. So parity's not really good enough, but it is where we're at the moment in at least much of our leadership positions, which we're very pleased about. We did the deep dive, the comprehensive analysis, and sort of out of this came what you can imagine: new policies, promotion strategies, recruitment strategies. The gender representation in sort of all our decision-making, communication, professional development, all sorts of things. Funding opportunities we instigated, travel grants, parental leave support, fellowships, that sort of thing. They've also all been implemented. We've made changes to parental leave to be best practice. Embarrassingly, they were not. And also improving access to parental leave for men, as well as women, to ensure all new parents have access to 14 weeks paid leave. These are the sorts of barriers. With all the goodwill in the world, there's so many practical barriers to women succeeding in the workplace, especially in the nature of the research workplace. So, as I said, we've achieved gender equity in our board, close to amongst our senior leaders in research and health, and our deputy leaders and our executives. There's still significant challenge in the Institute in some particular areas, but we're well on the way. We've achieved an accreditation with this Athena Swan group, which is well known to some of you that follow the academic world of this push to be more equitable with respect to gender, and it's a sort of formalized process, quite a tough process that you voluntarily submit yourself to, and it's been fantastic for us. Basically, it's a process where you get judged on improvement, not on the numbers that I just gave, parity, and obviously that's important, but it's you keep getting judged on whether you're progressing, and I really like that nature, that element to the Athena Swan award scheme that we've signed up so enthusiastically to. So, I am, from initially being deflated to where we were, given the nature of our organisation, I'm really proud of where we've gotten to. Conscious that there are still so many challenges, and I know that there'll be many of you who work at the Burnet sitting in the audience going, Brendan, we're far from perfect, and I absolutely agree with that, but I can assure you that we're incredibly determined to keep improving, to see this through as an organisation internally. As I say, clearly the nature of our work, I think, is much less of a concern for us, but we've got to live that as an organisation as well. And I think we are on our way, thanks to so many of the people who are on this call and their predecessors. And I'm always very open to feedback as how we can do things better. There's no greater challenge for us. There's no greater challenge for our organisation, for our society, for our region, than the one that is we're facing today. There are other big equity issues, and I mentioned one of them at the start of this talk, but there's nothing greater than this. We recognise that and put it front and centre in how we measure the success of this organisation. So thank you so much for having me today. I do feel quite privileged to be here, and Jen, I hand back to you and look forward to the rest. Thanks.
Wonderful. Thank you so much, Brendan. On to our speakers for the day. So today you will hear from Professor Gilda Tachedjian, Dr. Lindi Masson, and two early career researchers, Brianna Jesaveluk and Jenna Wilson. And the topic today is unlocking the vaginal microbiome to reduce the risk of HIV and preterm birth. And we chose this research to highlight in these speakers because it's an area of research at Burnet that's dominated by female powerhouses and is really world-class research that's being done for women by women. So speaking first will be Gilda. Gilda is the head of our life science at Burnet and the head of the retroviral biology and antiviral lab. And she's been at Burnet for a mere 21 years. She's a molecular virologist working to identify and develop antiviral molecules that prevent and control emerging viral pathogens such as HIV. She's made major contributions to a body of research for HIV reverse transcriptase, antivirals, drug resistance, and HIV prevention. She's also a pioneer in the study of genital microbiota and is deepening our understanding of the protective properties of lactic acid. And if you're not sure what that means, don't worry, Gilda's here to explain it now. So over to you, Gilda.
Thanks so much, Jen, for that kind introduction. Can everyone hear me? I assume they can. Look, I'm just going to clarify. I've actually clocked over 31 years of discontinuous service at the Burnet. It was actually broken spending 4.5 years completing my postdoctoral studies at Columbia University in New York. So I've actually had the unique opportunity to work under the leadership of our 4 Burnet Institute directors. So there's Ian Gust, our founding director, John Mills, who was also my PhD advisor. And I have to say, if it wasn't for his support and encouragement, I wouldn't be where I am today. So many thanks to John. Of course, Steve Wesseling, when I came back from New York, he was director. And our current director, Brendan Crabb. So thank you for your support. So, I think it's a competition. I'll probably beat the competition, Brendan. You've clocked over 15 years. Now, before you all start doing sums in your head to figure out how old I am, I've probably got another 5 to 7 years to go. So I'm just getting started to unlock the secrets of the vaginal microbiome to transform women's sexual and reproductive health with collaboration with my terrific colleagues who you're going to hear from today. So before I continue, I also like to acknowledge and pay my respects to the Bunurong people of the Kulin Nation, who are the traditional custodians of the land where I'm located. I'm at Burnet HQ in Melbourne, and I'd like to pay my respects to their elders past, present and emerging. So I'm going to share my slides. There's going to be a bit of technical things going on here before I get it right. So the wonders of Zoom. And I have to say that I gave a live talk a couple of weeks ago, and I do prefer it, even though it's more nerve-wracking to actually do it live because I can see people's faces. All right, so do we have the right aspect here? Yep.
Yes, it's good.
Fantastic. All right. Now at the Burnet, and I hope I can move this. Yeah, at the Burnet, we're always thinking about how our work is going to impact key populations. And so the key population that our work is aiming to target, and this is work by myself and Lindi, we were thinking about this over 10 years ago or more, is to target women who comprise 49% of the 1.5 million new HIV infections in 2021, with the majority of these infections occurring in adolescent girls and young women in sub-Saharan Africa, who are 2 to 3 times more likely to become infected with HIV compared to their male counterparts. So there it goes. So if we look at our region, in Papua New Guinea, women have higher HIV incidence than men. And while the numbers are obviously lower in Australia, just recent figures, 40% of new HIV infections are due to heterosexual sex are in women. So HIV is still an issue in women. So I'm going to talk about vaginas, and I think this is the appropriate audience. So most HIV infections actually occur by the virus entering through the lower female reproductive tract, where the virus can establish infection by penetrating this thick layer of the vagina and the ectocervix, as well as penetrating this thinner layer of the endocervix. But the probability of HIV transmission is usually low compared to other forms of transmission, such as parenteral or transmission through blood. And that's because the vagina is typically a formidable barrier to HIV infection. And I'll tell you why. And that's because of the physical barriers. So we tend to have mucus sitting on top of the endocervix that acts as a physical barrier, as well as the epithelial cells of the ectocervix vagina and the vagina. And this not only acts as a physical barrier to the virus penetrating, but also it acts as an immunological barrier. And these epithelial cells respond to the microbes, and they produce immune mediators that have inflammatory activity or response. And that inflammation is typically important for clearing infections, although paradoxically for HIV, it actually increases HIV acquisition. And then colonizing the surface of the epithelium is the microbiome, and in particular, the beneficial lactobacillus species that can work in a variety of ways to prevent HIV. A lot of these have been worked out in vitro, but not in women. And that includes a byproduct or metabolite that lactobacillus species make, which is called lactic acid, which I'm going to talk about later. But I thought I'd just segue into what is the microbiome. So the microbiome are communities of bacteria, along with viruses and fungi that inhabit different regions of the body. So we've got the gut microbiome, the skin microbiome in the mouth, of course the vagina, and there's even a penile microbiome. And so this microbiome is distinct within an individual. So if you look at different parts of the body, you can see the composition is different. It can change with time. So I think we all know perhaps the gut microbiome will change with diet, and it does differ between individuals. And it's been said that there are more microbes than there are stars in the Milky Way. And so there are 100 trillion microbes that inhabit our body. And in fact, there's more microbes than there are human cells. So perhaps we consider ourselves more as microbes than as humans. And 95% of the microbiome is actually located in the gastrointestinal tract. And so it's probably not a surprise that a lot of the research that has been done so far has been focused on the gut microbiome. And we know that the gut microbiome is essential for health. So you'll have optimal gut microbiota that is needed for our survival. So producing nutrients in the gut, such as vitamins, important for the development and maintenance of the host immune system. There's also a gut-brain axis as well. So it's important for our mental health. And as well, these essential microbiota protect against pathogens. And here's a lovely image of rod-shaped bacteria that inhabit our feces. So there you go. So the microbiota can also be harmful. There can be an imbalance in the microbiome. And this imbalance is associated with a variety of human diseases. There's a myriad here that's listed. This is an association, may not necessarily be causation, but quite interesting. So increased risk of, say, cardiovascular disease, diabetes, obesity, just to name a few. Now, like the gut microbiome, the vaginal microbiome comes in different flavors as well. It can be harmful, which we call non-optimal, or beneficial. And so the non-optimal vaginal microbiome is characterized by this increase in diverse anaerobic bacteria and a depletion of the beneficial lactobacillus species. And the most common clinical manifestation of the microbiome or non-optimal microbiome is called bacterial vaginosis, or BV, and this occurs in women of reproductive age. And it's typically diagnosed by a Gram stain called a Nugent stain, or via clinical criteria such as the Amsel criteria that includes an abnormal vaginal discharge. And if we look at women in South Africa, up to 50% of these women have BV where HIV is prevalent. And if we look in our region, and the data isn't extensive, but there are a couple of studies that show that 20 to 30% of women of reproductive age in Indigenous populations in Australia and in PNG have bacterial vaginosis. So why do we even want to care about BV? Well, it actually increases a woman's risk of acquiring and transmitting HIV to their male partners. It also decreases the efficacy of topical pre-exposure prophylaxis. So like a microbicide women can use to prevent HIV infection from their male partners. And this is all in contrast to an optimal vaginal microbiota that is dominated by these lovely, juicy lactobacillus species, which is associated with favourable sexual and reproductive health outcomes, including protection against HIV. So in 2019, a group of 18 experts, including myself and Lindi, got together to write a consensus paper to standardise the nomenclature surrounding the bacterial vaginosis, as well as the microbiome in the field. And I have to say, this came off a meeting, a very high profile HIV prevention meeting that was held several years ago. And I was at this symposium and there was a heated discussion about how to define BV. So we thought, oh, well, we need to come up and write this consensus paper. And so what we landed on as a group is that non-optimal microbiota should be referred to in non-stigmatizing language. So if we're talking about BV, instead of saying unhealthy or abnormal, we mention, or we use the word non-optimal. And then we've defined non-optimal microbiome based on the impact on the host, so not necessarily the composition of the microbiome. So that microbiome elicits a genital inflammation and or is associated with adverse sexual and reproductive health outcomes. And we also specified the method used to determine BV, whether that's a very basic Gram stain, Nugent's Gram stain, or the clinical criteria, or now there's these molecular methods that have come online, such as next-gen sequencing, that has given us a better way of, or definition of looking at resolution, looking at the composition of the microbiome. And that's illustrated in this study in young, healthy South African women, where they use what's called 16S ribosome RNA gene sequencing to look at the composition of the microbiome, or the vaginal microbiome, in these women. And each vertical line represents a woman and the colours represent different bacteria. So only 10% of these young women in Africa had lactobacillus crispatus dominated microbiota, and this is considered beneficial. 32%, and that's with the green, have lactobacillus inners. Now, it is lactobacillus, but I call I always say that, you know, not all lactobacillus species are created equal, and L. iners is actually not as stable as L. crispatus and transitions into a non-optimal microbiota. And you can see here that 58% of these women have what we consider non-optimal, where there is a depletion of beneficial lactobacillus species. And in this group here called CT3, you can see there's a lot of blue bacteria, which is G. vaginalis, which is normally associated with BV. And then this group here, CT4, where there's this highly diverse vaginal microbiome. If you can see that where there's G. vaginalis, there's things called Sneathia and Megasphaera and all these nasty anaerobic bacteria. So this was a longitudinal study. And so at the beginning, these young women were HIV negative, and they were followed with time. And what they found was the women with CT4, the highly diverse vaginal microbiota, had a fourfold or greater than fourfold increased risk of acquiring HIV compared to women with the L. crispatus dominated microbiota. And another important finding of this study is that women who had vaginal microbiota depleted of lactobacillus species, so CT3 and 4, had high levels or elevated levels of these pro-inflammatory cytokines and chemokines. It's increased genital inflammation. And I'm going to give a plug to Lindi, because Lindi was one of the first to actually report high levels of genital inflammation associated with increased risk of HIV. She could correct me if I'm wrong. So why is this genital inflammation a problem? So this subclinical genital inflammation results in recruitment and activation of the HIV target cells. So this is the vagina, and here you can see the CD4 positive T cells, and with inflammation, these are activated, and then they move further to the surface of the vagina, so that these little particles, HIV particles, have an easier job in infecting them. And then with BV, you also see a disruption of the epithelial barrier integrity. See these gaps? The virus can now penetrate those gaps to infect the target cells. And then these BV bacteria produce a lot of enzymes that can also break down the mucus barrier. So this is in contrast, if you've got a non-optimal microbiota, this is in contrast to an optimal microbiome, lactobacillus dominated, where you have a non-inflammatory environment, you've got an intact epithelial barrier, and this reduces HIV risk. So over the last 10 years or so, we've been asking the question, how do vaginal lactobacilli protect against HIV? So the two distinguishing features between women with optimal versus non-optimal microbiota is the pH. So the pH in women with BV is elevated to greater than 4.5. The second distinguishing feature is the concentrations of these metabolites, and in particularly lactic acid produced by lactobacillus species, which you've got fairly high levels in women with an optimal microbiota, but it's depleted in women with BV. And that suggested to us at the time that there's something going on with lactic acid and may have a protective role in the female reproductive tract. So this is lactic acid, a very simple molecule and pretty cheap to purchase too. And so this molecule is responsible for driving the pH in the vagina to low levels, as low as pH 3.8. And just an interesting fact, that women are the only species that have acidic vaginas. Not even the non-human primate comes close with regard to the vaginal pH. So studies have shown that lactic acid at physiological concentrations can kill the BV associated bacteria, but not the vaginal lactobacillus species. We've shown at the Burnet that lactic acid potently kills HIV, and others have shown that it kills other sexually transmitted infections such as chlamydia and gonorrhea. And we've shown here that lactic acid has direct anti-inflammatory activity on the epithelial cells from the female reproductive tract. So that could drive down genital inflammation. And more recently, we've shown that lactic acid can actually strengthen the epithelial barrier of the female reproductive tract, which would make it difficult for HIV and other STIs to penetrate and establish infection. Now, I just wanted to highlight this paper that we published in 2017 in the journal Mucosal Immunology, a diverse group of researchers and a great collaboration with Anna Herbst, who unfortunately is not here. This is today, Wednesday is her day that she works. She doesn't work, unfortunately. So Anna and individuals from my lab and collaborations with Rafi at the Burnet and collaborators at Boston University. And so in this study, we showed that lactic acid has direct anti-inflammatory effects on cervical vaginal epithelial cells. And I highlight this paper because this work resulted in awarding of a US patent that was granted to myself and Anna Herbst as co-inventors. And this patent covers the ability of lactic acid to decrease genital inflammation of any condition in the female reproductive tract, including bacterial vaginoses. And another study I want to highlight was published last year in the prestigious journal Microbiome. Here we show that lactic acid is able to enhance the epithelial barrier integrity of the cells in the cervicovagina. And this was a study that you can see it takes a village, there's a lot of people, and was the efforts of 2 consecutive PhD students, David Delgado Diaz, who's finished, and Bree, who's going to be speaking today. And it was a great collaboration with Lindi, who was recently recruited to the Burnet Institute. And Lindi has access to relevant cohorts in South Africa where she can take vaginal samples and then analyse them with a really cool technique so that she can look at the host proteins and the micro proteins. And just a long story short, she was able to add to this study by providing evidence that in these women, lactic acid is associated with this increase in barrier integrity in the context of these women. And I'll put this link here because this journal put together a fantastic video abstract that summarises this work to a lay audience. So if you have time, I really urge you to have a look at this video. So like the gut microbiome, lactobacilli in the vagina need a food source. So in this case, it's glycogen, which is broken down so they can use it. And then we've got our lactobacillus species. And we could technically use lactobacilli to optimize the vaginal microbiome and environment. And indeed, people are looking at that strategy as a live biotherapeutic approach, or even a vaginal microbiome transplant, sort of taking from the gut microbiome field with the fecal microbiome transplants. And so you could perhaps colonize the vaginal microbiome with these live biotherapeutics, and these lactobacilli have a variety of important properties listed here. And I just mention this because Lindy's group is looking at live biotherapeutics approach, and Jenna will be talking about that today. But we in my lab have been focusing on the metabolite that is produced by the lactobacilli, and we've shown that this metabolite, at least in vitro, does recapitulate a lot of the properties of the live lactobacilli. And I have to say that, these approaches are not mutually exclusive. We could technically combine these approaches to come up with a more effective way to optimize the vaginal microbiome and environment. But as I said, we've shown this in vitro and ex vivo, so we need to show this in women. Because people aren't going to invest in lactic acid to develop it as a drug. As it turns out, there are lactic acid containing products on the market which have apparently have an impact on bacterial vaginoses. But these products are over-the-counter gels that haven't gone through the rigorous regulatory pathway that normally you would have for a drug. And in collaboration with our partners or colleagues at Melbourne Sexual Health Centre, including led by Katrina Bradshaw, they showed in this systematic review that high quality evidence is lacking to show that these products do anything with regard to the microbiome or even modulate the vaginal microbiome, and that we need adequately powered, rigorous randomized trials to prove this. And none of these studies looked at the inflammatory markers and epithelial barrier markers that we're interested in to show. And Bree is going to talk to you about limitations of using the over-the-counter lactic acid gels. And we've actually ended up formulating our own custom-made lactic acid gels, which we're going to go ahead into a clinical trial, funding permitting. And this is a mechanistic study, and it is randomized, double-blind, placebo-controlled in collaboration with Katrina Bradshaw, Melbourne Sexual Health Centre, where we can test the lactic acid gels against placebo in women who dose this gel for 7 days, and then look at the outcomes. And all these outcomes, looking at the microbiome, genital inflammation, epithelial barrier, powered to actually find a significant difference. And so this year, we're completing preclinical studies. I probably don't have time to go through this, but with help from Burnet, who's partly funding some of these studies. We've started with the gel formulations, the R&D gel formulations, which will go through quality control and various other tests, and we'll get our ethics done so that we can start the trial next year with GMP clinical batches. And I'm going to just do a side note here that we actually submitted this proposal to NHMRC last year, and it got a category 6 out of 7. In the old days, that would be automatically funded. So we just missed out. So we're going to go in again. So how can we use lactic acid to prevent HIV? So if we can show in our mechanistic study that lactic acid does all the things that we saw in vitro, then that will promote investment in developing sustained release modalities for lactic acid, such as an intravaginal ring to release lactic acid in a consistent way. And that could be used as an adjunct with topical HIV prevention technologies to maximize the efficacy of topical pre-exposure prophylaxis. Our gel can also be used. So just as a side note here that with bacterial vaginoses, the standard of care is a course of antibiotics, but the recurrence rate of BV is really high, 60% in over 6 to 12 months. So where we could use our gel is as a periodic presumptive treatment. So women would be treated with antibiotics, but then you would periodically use the gel, say perhaps after menses, to optimize the microbiome to help prevent that transition to bacterial vaginoses. And then the non-optimal microbiome is actually associated with associated with infertility, increased risk of STIs, spontaneous preterm birth, and miscarriage. So there's a lot of things that we can do. We can optimize the microbiome, and Lindy's going to be talking about looking at biomarkers in vaginal fluid. She's developed a lovely point of care test to predict whether women are at increased risk for HIV and STIs. And we're collaborating with investigators, an investigator in the University of Western Australia to look at biomarkers for spontaneous preterm birth and predicting this, being able to predict this in Indigenous populations in Australia. So just finally, I hope that you sort of appreciated that we've got this dynamic team of researchers, myself, Lindy and Anna Herbst, who are really have expertise that can move this genital microbiome field forward. And we've come together with the help of Serena Kukutsa from the business development team to develop this genital microbiome initiative. And so it's in the early stages, but we have come up with our vision to transform women's sexual and reproductive health in Australia and globally. Our purpose is to create and translate knowledge on the genital microbiome into better sexual and reproductive health outcomes for adolescent girls and young women. And we've defined our objectives to define the role of the genital microbiome in Australian First Nation women and women globally, including Indo-Pacific, to identify host and microbial biomarkers to translate into low-cost diagnostics, and this is with the help of the BDI, to predict adverse sexual and reproductive health outcomes. And Lindy's we'll talk about the GIFT device as well. Identify bioactives and small molecules as well as live biotherapeutics to translate these into novel therapeutics and prophylactics, and to collaborate, with key populations to promote health literacy on the role of the genital microbiome in health and disease for advancing into novel interventions through co-design. And this is with our public health, in collaboration with our public health researchers here at the Burnet. Co-design is really important because you can make any intervention, but if the end user can't use it or doesn't think it's appropriate, then it won't get used. And with that, I'd like to acknowledge everyone on this slide. It takes a village to do this work, and our funders. And I'll stop there to give Bri a chance to talk. So I'll just now, if that's okay. We take questions at the end, right? So I think now it's my pleasure to introduce Brianna Jesaveluk, who's a third-year PhD student in my lab. She started off doing honours in my lab on bat research, but she had her eye on the vaginal microbiome work. So as soon as she got her PhD scholarship, she wanted to work on the microbiome, and she has a passion for translational research. So Bri's going to present our surprising findings on over-the-counter lactic acid-containing gels and showing how our bespoke gels are better. So over to you, Bri.
Thanks, Gilda. Do you mind just stopping your share?
Oh, okay. Will do. Sorry.
No problem. Hi, everybody. Just one moment. Oh, sorry, just one sec. Is that all displaying correctly?
Yeah.
Thank you. Okay. Happy International Women's Day, everyone. Thank you so much for taking the time and celebrating with us. So as Gilda said, I'm Brianna and I'm her PhD student in her lab. So I'm excited to tell you a bit more about the work that we're doing with vaginal gels, including studying the limitations of existing over-the-counter gels on the market and some of the preclinical data that we've gained with testing our bespoke lactic acid gels. And sorry, there we go. Okay. So the work that I'm presenting was published last year, and it was a collaboration between researchers at the Burnet, as well as experts from around Victoria and overseas. So Anna, David, and of course, Lindi are at the Burnet, and the Simons are materials science experts based at Swinburne, and they helped us with developing the testing methods that we used. Katrina and Jacques are leaders in the field of bacterial vaginosis and the vaginal microbiome, and they assisted us with this work. And this was done in collaboration with Sayum Ayahuni at the MatTech Corporation in the US, and they're a company that produces the epivaginal tissues that we use to test the gels, which you'll see on the slide here. I'll explain a bit more about the tissue model shortly, but just first, I'll give you some background on the over-the-counter gels. So on this slide are some examples of vaginal gels that can be found on the market around the world. They're usually advertised to treat bacterial vaginosis or to restore abnormal vaginal symptoms like discomfort and odor and vaginal discharge. The gels shown here all contain lactic acid amongst their various ingredients. You may be wondering about the availability of gels in Australia. To my knowledge, there are less options available over the counter in Australian chemists. This Acid gel is a similar product in Australia, but it doesn't actually contain lactic acid. These days, it's easier to source these products online, and new products are entering the market all the time. However, as Gilda highlighted, there is a lack of quality evidence showing that these products work as treatments for BV or for other vaginal symptoms. So as well as conducting the systematic review of the published evidence that Gilda pointed to, we wanted to test some of the gels ourselves to see what effect they have on vaginal cells. So we ordered them into the lab and we started our testing. And the model that we use to test these gels is called the epivaginal model, and it has a similar structure to human vaginal tissue. So it has a 3D structure with differentiated cell layers, just like in a woman. And this means that we can test our gels accurately without needing to use animals before we go on to test them in women. In fact, although a rabbit vaginal irrigation model is usually used to test the safety of vaginal products, using the 3D tissue model is more sensitive and accurate, as Gilda pointed to. There's limitations to using animal models for testing vaginal products. Plus, it does spare the bunnies. So here we have the data which shows how well the cells in this model can survive when treated with the various over-the-counter gels. So this is compared relative to the cells that aren't treated. And you can see that for the majority of these gels, they appear to be very toxic to the vaginal cells. Compare this now to our bespoke gel, which is shown in blue, and that toxicity effect is greatly reduced. You can see that one of the over-the-counter gels, called Restore, also seems to be less toxic than the others. However, we also looked at the effect of the gels in producing an inflammatory response, which includes cell markers, which are called cytokines, including IL-6 and IL-8. And although Restore was not associated with toxicity, it did cause a large spike in the cellular stress response. So you can see that this was not the case for our bespoke gels. In summary, all of the gels which contain lactic acid either kill all the vaginal cells or they cause a big inflammatory response. And our bespoke gel does neither, which is exciting. The next data I'm showing you was generated using our Burnet anaerobic chamber, which you'll learn a bit more about in our upcoming video. In it, we can grow bacteria, which are isolated from the vagina. And we wanted to test whether these bacteria could grow in the presence of lactic acid containing gels. And we found that our bespoke gel is able to kill the bad bugs, or G. vaginalis, while not affecting the viability of optimal bacteria, so that lactobacillus crispatus that Gilda mentioned. We found that a placebo gel didn't affect the bacterial growth, and so this active killing was likely due to the lactic acid in our gels. And this could be a great sign for using these gels as a better and more permanent treatment for BV than antibiotics, since we're encouraging the survival of only the most optimal bacteria. So to summarize what I've shown, we found that our bespoke lactic acid gels are safe in the vaginal model, and they don't cause cell death or inflammation. This is in contrast to the gels that are already on the market, which can cause toxicity and a pro-inflammatory response. Some more data from the publication, which I haven't gotten into today, in fact shows that our gel formulation elicits an anti-inflammatory response in the cells. Lastly, our gels actively kill harmful bacteria, but they don't kill beneficial lactobacilli. So we can't wait for the opportunity to test these gels in a pilot study in women so that we can learn more about their potential benefits for treating BV. Thanks very much.
Thank you so much to Gilda and Brianna. I think we can all agree that is an amazing body of work, and they're just getting started. It's so exciting. And I must share that I am particularly inspired by their work for a number of reasons. Obviously, I'm a woman and directly affected by the research, but I was also trained as a molecular microbiologist. So looking at all the different species in the microbiome, I find fascinating. Working in translational research at the moment, I can see such potential for multiple products to come out of this work that we'll be able to see actually help women. And as a side note, my job before Burnet, I was the CEO of a startup that had a medical device for pelvic floor recovery after birth to stop incontinence. And so I was doing a lot of pitching to venture capitalists that were predominantly men. And if I ever said vagina, the entire room freaked out and looked away. And so just the sheer number of times in that presentation, Gilda, that you could say vagina was really refreshing and wonderful to see. So I think we are just on time, so we can probably take a couple of questions now, if anyone has any for Gilda and Brianna, before we move on to Lindi, sorry. Are there any questions? Anyone? Yes, Alison. Just unmute. Still muted, Alison. Yep. Are you able to unmute as host? There we go.
Can you hear me now? Yeah, okay.
Yes.
No, look, that was most informative. So thank you for that. I suppose the just one high level question is, what's the expected time frame to be able to go through the testing of the gel that you're working on, and then maybe, you know, move it to a commercialization stage? And have you got enough funding, immediate funding to do that?
Well, I'll start off with the last question. There's never enough funding. So, but in terms of immediate funding, so the plan is to do the preclinical work on the lactic acid gels, the R&D. So we've got that all mapped out for this year, and there's some support from Burnet. We've put some VMRAF funding in, submitted that to help support that. And then we are applying for an NHMRC grant to support the mechanistic study. And I think you probably know that we applied for an MRFF stage 2 grant. And one of the comments was that we needed to do this clinical study to show that lactic acid actually does all the things that we showed in the test tube. And so that's the next step, to try and get that funding. Now, if we get that funding, then the timeline would be, it's 1.5 years to actually do the clinical studies, so recruiting women and getting the samples, and then the analysis another year or so. So that will take time, but we are working with Serena and business development, where we'll have to go and approach, you know, companies like Bayer and so on, to try and attract funding to see if they're interested in supporting this. And then the next stage, you know, if we show there's an effect there in that context, would be for the gel, would be to do another study, which is that periodic presumptive treatment that I talked about, where you would have a study, and this would have to be a multinational study, where you would have women take standard of care, which is the antibiotics, and then you try that periodic presumptive treatment with the gel to try and prevent BV recurrence. So we've done that market analysis to show that BV recurrence is an unmet need. And then there is a need to get away from antibiotics and antibiotic sparing type treatments because of antimicrobial resistance and so on. So in terms of time, things take a while, Alison. So anyway, I hope I've mapped that out for you.
Thank you, Gilda. I was just curious because it seems that you've done you have done a tremendous amount of work. I was just curious as to, how much further you have to go. But as you said, you've got it all mapped out. So thank you.
Thanks for the question.
Gilda, can I just read out one pre-submitted question for you before we move on? Margaret submitted, with hormonal fluctuations in some women, how does this affect the vaginal microbiome and its defense against HIV or STIs?
That's a brilliant question. So there is data showing that hormones can actually have some sort of impact on the cytokines and chemokines that are released. And then there are, hormonal contraceptives, some of which actually shift the microbiome to BV. Now, with our gel, we've been doing some tests to make sure that the activity of lactic acid, in particularly the anti-inflammatory effects, are still observed in the presence of female sex hormones, as well as hormones that are used for contraception. And so far, this was ACH2 funded grant, and so far we haven't seen any adverse impacts on the activity of lactic acid in that system. So that looks, pretty promising at the moment. Yeah, promising at the moment.
Great. Well, in the interest of time, if you have further questions, we'll hold them until the end. But now it's my pleasure to introduce Dr. Lindi Masson. Lindi has been involved in HIV and genital microbiome and immunology research for over 10 years. She is currently leading 2 clinical cohort studies and a translational research study in South Africa. The primary focus of this research is to understand the socio-behavioral and biological factors associated with HIV acquisition risk in South African women and develop tools to reduce this risk. An exciting and emerging area of work, which she'll speak about today, aims to address non-optimal microbiome and the resultant adverse maternal, sexual, and reproductive outcomes in Australian women. So over to you, Lindi.
Thank you, Jen. Yeah. Hi, good morning, everyone. Thanks so much for this chance to showcase some of the research that we're doing in my group and with our collaborators in South Africa. So I'm going to switch topics a bit and talk about diagnostics. The project that I'm going to speak about is called GIFT, and it stands for Genital Inflammation Test. And this is a screening test that we're hoping will help to reduce the prevalence of sexually transmitted infections and bacterial vaginosis in women in resource-limited settings, and in that way, reduce their risk of HIV infection and improve their reproductive health. So I'd like to start with this very busy acknowledgement slide. As Gilda said, it takes a village. There are a huge number of collaborators involved in this work. I'd like to firstly, and most importantly, acknowledge the participants of the 5 cohort studies, Caprisa, FemPrep, YouChoose, Wish, and MISC, who have played an absolutely critical role in all of the work that we've done to date. I'd like to acknowledge the traditional custodians of the lands on which I'm presenting from today, the Gundungurra and Dharawal people. I'm up in New South Wales at the moment. And I'd like to acknowledge our core GIFT Africa consortium that's led by Professor Joanne Passmore at the University of Cape Town and myself at the Burnet, and also our other external collaborators and our funders. So sexually transmitted infections and bacterial vaginosis, which Gilda very nicely introduced during her talk, are highly, highly prevalent in African countries and also in other resource-limited settings. These infections and conditions cause high levels of inflammation in the female reproductive tract, and this inflammation is associated with very high risk of HIV acquisition and also a high risk of reproductive complications like preterm birth. So at the moment, in resource-limited settings, the STIs and bacterial vaginosis are managed syndromically. So this means that they're only treated if women have symptoms. However, some of our research has found that the majority of women, over 80% in some studies, who have these STIs or BV actually don't have symptoms. So one might think that maybe by addressing the symptomatic women, you're treating the more severe infections. However, we've also shown that women who don't have any symptoms have the same level of inflammation in their reproductive tracts as women who do have symptoms. And so this means that they're at equivalent risk of HIV infection and reproductive complications as symptomatic women. So we really, really need to develop tools to address these asymptomatic infections and underlying inflammation. So I'll tell you a bit more about syndromic management. It was recommended by the WHO, the World Health Organization, in the early 1990s. And it means that patients are given a range of antibiotics based on what the clinician thinks the woman has. So these antibiotics can treat a number of underlying infections. And it was implemented for very good reasons. So it's inexpensive, it's easy to implement, you don't need specialized labs, staff, equipment, and patients are given immediate treatment. And this is something that's very important, particularly in resource-limited settings, because return rates are very low. So women often live far away from the clinics, they don't have a lot of money for public transport, and there are really long wait times at clinics. So they're quite unlikely to actually come back to a clinic for test results and treatment. But the cons are, as I mentioned earlier, the majority of women don't have symptoms. And then by just presumptively providing antibiotics, there's a lot of overtreatment. So women are treated for infections that they actually don't have. So this nice modeling study was done by a collaborator at the University of Cape Town, and it was based in South Africa. And they looked at how the prevalence of different sexually transmitted infections, bacterial vaginosis, and candidiasis changed after syndromic management was introduced. And they found that while there was a substantial reduction in ulcerative infections that can be cured, like chancroid and syphilis, there was actually very little impact on the discharge-causing infections, gonorrhea, chlamydia, trichomoniasis, as well as bacterial vaginosis and candidiasis. So we're kind of stuck with syndromic management, though, because we actually there aren't low-cost alternatives for most STIs and BV, or a way to detect vaginal inflammation. So that brings us to the aim of our work. So we want to try and reduce the risk of HIV and improve reproductive health in women by identifying by developing a tool to identify vaginal inflammation that's caused by discharge-causing sexually transmitted infections and bacterial vaginosis. So the infections that syndromic management really just has not had an impact on. So we started this work in 2010 during my PhD in Joanne Passmore's group. And it's been a fairly long road, starting with problem identification, the discovery of biomarkers, validating the biomarkers, developing a test, conducting cost effectiveness analyses. And now we're at the point where we're about to manufacture this month, actually, and we're going to be enrolling a large cohort study in Zimbabwe, South Africa, and Madagascar this year. And at the same time, we're conducting user experience studies. As Gilda mentioned in her talk, these are really critical. There's no point developing an intervention if the women aren't going to use it. And then after that, we also have funding for a registration trial and pre-market testing. So this has led to a number of publications, European and South African patents, and our clinical validation study is now registered on clinicaltrials.gov. And our overall aim, this is where we currently see the positioning of the gift device, is that it could be offered to any woman who are attending family planning, HIV pre-exposure prophylaxis clinics or antenatal clinics. It could be self-administered, so we're aiming for something that's very easy to use, or nurse-administered, and we're aiming to develop something that's rapid, so results within 20 minutes, low cost, equipment-free. And our hope is that we can identify these women with inflammation caused primarily by one of these discharge-causing infections or bacterial vaginosis. And we're working with partners at the Burnet Institute and the London School of Hygiene and Tropical Medicine to develop our management strategies, which will likely be context-specific. So this very busy graph is our biomarkers discovery data. So we measured a whole range of markers of inflammation in vaginal samples from South African women. And we also tested them for sexually transmitted infections and bacterial vaginosis. And so these stacked bars here show the level of inflammation. So the higher the bars, the more inflammation the women have. So you can see here that all of these infections increase the level of inflammation, although some were more inflammatory than others, like chlamydia was the most inflammatory, and bacterial vaginosis was associated with a bit of a mixed profile where some inflammatory markers were increased and some were lower. So we used this data to narrow down to 3 different markers of inflammation that were the most accurate combination of biomarkers. And in this cohort, we found that in HIV negative women, the biomarkers correctly classified 76% of women. So they identified 72% of the women who had an infection or BV. In HIV positive women, the biomarkers performed a bit better. So 84% of women were correctly classified, 80% of women with an STI or BV were identified. And compared to the current standard of care, clinical signs and symptoms, this was a vast improvement. So signs and symptoms identified only 19% of women, missing 81% of the women who had an infection in this cohort. And importantly, we were able to detect 82% of the women who had vaginal inflammation. So we then went on to validate these biomarkers in different cohorts in different regions of Africa. And we found that the biomarkers performed really consistently across the different cohorts and across a whole range of different vaginal sample types that we looked at. And they were consistently better than the current standard of care, signs and symptoms. We also found that if we also measured vaginal pH, which Gilda introduced really nicely, it's a marker of an optimal versus a non-optimal microbiome, then we could correctly classify 82% of women with one inflammatory marker and vaginal pH. And most importantly, we found that a vaginal swab performed the best. So this is a sample that's easy to collect, can be self-collected, and this is really important if we're looking at potential self-collecting, self-administration of this screening test. So this image here is our first prototype that we've been working on optimizing, and we're currently working on our second prototype that looks a little bit different to this cartridge, but the principle remains the same. So this is what's inside, or it would be very similar to what's inside the COVID rapid test that I think most people are very used to now. You apply the sample into the sample reservoir, so there's a sample pad underneath, and then the sample runs or flows along this nitrocellulose strip. And while it flows along, it picks up antibodies for whatever you're trying to measure. So in this case, our inflammatory markers. And then the inflammatory markers then bound to the test line. And the test line intensity gets compared to a printed reference line. So you can see if it's higher than the reference line, then it's a positive test. And if it's lower than the reference line, then it's a negative test. So this is the basic principle that we're working on at the moment, combining 2 of the inflammatory markers into one line, and we need a separate test for the third inflammatory marker. Although we're going to be evaluating this in the clinical study, and there's potential to modify this and drop 1 or more markers. So this means that for each biomarker, we need 2 different antibodies. And this meant that we needed to screen, I think, about 1,500 different combinations of antibodies to identify the best pairs, because they need to work well together as well. So we also had to screen different buffers, different quality controls, and we screened different pooled clinical samples and individual clinical samples as well. And this is just probably a fraction of one day's work that these 2 lovely ladies did in Cape Town. Mona Lisa Manhanja is a postdoc in my group, and Fazila Kumalo is part of our point of care development team. So a huge amount of work led to the identification of 2 really good pairs for the one biomarker and 4 pairs for each of the other 2 biomarkers. And this is an example of one of our pre-manufactured devices that we're currently evaluating at the moment. So you have a control line and then a test line. And then the test line currently gets compared to this gold scorecard. So the intensity of the line gets graded based on the scorecard because the reference line has not yet been integrated. And so using this system, you can see here we've evaluated our controls and then pooled clinical samples. And basically for all 3 biomarkers, the negative controls and the negative pools are negative on the device, and the positive controls and the positive pooled clinical samples. So when I say pool, I mean multiple samples from different women all pooled together. Then we see nice positive bands. And then when we look at the individual clinical samples, then you can see in general an increase in the gold score as the biomarker concentration increases. So as you have a higher concentration, then the band becomes darker and darker, which is really important. There are some exceptions. This one, sample number 2, for example, that we're currently working on optimizing and tweaking. So alongside these activities, we've also been conducting other analyses. This is our first cost effectiveness analysis that was done in partnership with the Health Economics Unit at the University of Cape Town. This is an analysis based in South Africa. And in the next stage of our project, this will be expanded to include different African countries, and also it will be expanded to include a more detailed analysis. But basically what you can see here, the incremental cost effectiveness ratio, it's good when it's low, but when it's high, it means it's very costly for how effective it is. The ratio is the lowest for the gift device, and it's much lower compared to other currently available diagnostics. Well, diagnostics that are available in first world settings anyway, but are not implemented in resource limited settings. So gift performs really well. There are some trade-offs though, because the specificity isn't 100%, then there is the risk of some overtreatment. And so those sorts of factors are going to be evaluated in more detail in future models. The next step that we've been working on, also in collaboration with other researchers at the London School of Hygiene and Tropical Medicine, was to conduct a Delphi survey to evaluate potential implementation strategies. So the surveys were administered to stakeholders in the field, and different individuals from all around the world really responded to the surveys. So in round 1, we had clinician, researcher, and policymaker respondents. And in round 2, we had primarily healthcare worker respondents, so clinicians, nurses, and also researchers as well. And so I've just picked out a few of the key questions that I wanted to showcase today. The interviews are obviously a lot more detailed than this, and we're currently preparing a manuscript detailing all of the findings. But the majority of respondents said that they would offer sexually active asymptomatic women testing with this gift device. And the majority also said, although this was a little bit more divided, the majority also said that screening with this device should be followed by diagnostic tests for to identify the specific infection before treatment. And then we looked at who we should be screening. And in general, all of the groups that were identified in the round 1 survey were also thought to be priority groups in the second round survey. And but the most agreement was with screening high-risk women of any age, in any place, in any setting. Then followed by women using HIV pre-exposure prophylaxis or using injection drugs. Then women of any age in high HIV prevalence settings, women attending family planning clinics, and women attending screening clinics for cervical cancer. So another place we want to evaluate the gift device is in the context of cervical cancer, because these biomarkers of inflammation might also be picking up women with cancer. And then we wanted to see if this device could be useful for women who have symptoms. And so apologies for this very detailed diagram, but this is the World Health Organization protocol, recommended management protocol for women who have a discharge causing or who have vaginal discharge. And there are 4 different streams, and the first 2 are recommended in settings where diagnostic assays are available for infections. And then 3 and 4 are the paths to follow when there's really very little available. So the take-home message here is that the respondents of the Delphi survey felt that the 3rd and 4th pathways were places where our screening tool could potentially be useful. So where a woman comes in, she has vaginal discharge, and they need to make a decision about whether to treat her, and if they're going to treat her, what antibiotics to give her. And so I also wanted to show this because I just love this graphic. This is our last meeting in summary. We had a graphic artist at the meeting who basically recorded the vibrant discussions and conversations throughout the meeting. This you can find on our website if you want to have a look at it more closely, but we basically have 5 different work packages: the management package, and then the device development work package, the clinical trial group, the integration group, and then the pre-commercialization group. And then very quickly, I wanted to just slot in some of our other work about how we're expanding on our biomarkers discovery and device development work. So this work includes a new method of measuring thousands of different proteins in any given biological sample. It's called metaproteomics. And we're using this to try and identify more infection-specific biomarkers. So our current biomarkers pick up inflammation, which is very useful, but we don't know what's causing it. So we're hoping that with this work, we can have a clearer idea of what the actual cause of inflammation is. So then we can give the woman immediate treatment without having to refer them for follow-on tests. So with this work, we've identified 2 proteins that identify women with bacterial vaginosis with 96% and 92% accuracy, which is really, really accurate. And we're in the process of looking at these in 2 separate cohorts of women. And then we'll be looking at filing a new patent for these biomarkers. We have lots of next steps. So this is very much a work in progress. We're almost ready to manufacture our device and evaluate it in our new cohorts. The new economic evaluation is underway. We're conducting user experience studies where actual nurses who are administering the device, as well as patients, are being interviewed about their preferences and whether or not they'd use the device, how acceptable it is, as well as other modeling studies and other user studies. And then after this, we'll be doing our pre-market testing and our registration trial. We'll also be validating the new biomarkers that are specific for BV. And Gilda briefly mentioned our exciting new collaboration with researchers in Australia to try and identify, using the methods that we've developed, to try and identify new biomarkers of preterm birth. So before I end off, I would like to quickly show a video that was developed by our team at the University of Cape Town. If it'll load nicely.
HIV is one of our most serious health challenges. The major drivers of HIV risk in women are undiagnosed sexually transmitted infections and bacterial vaginosis, both highly prevalent and recurrent in Africa.
In fact, more than 80% of women with these conditions do not get treated because they don't have obvious symptoms. Our team at the University of Cape Town have developed the genital inflammation test, GIFT, using a patented technology platform to accurately identify women who currently don't have access to treatment.
GIFT is true point of care, low cost, and easy to use. It is a screening tool that has 82% accuracy to detect STIs and bacterial vaginosis. This innovative diagnostic device will significantly improve case finding for conditions known to increase HIV risk in women. Our vision for GIFT is to make it available at every sexual and reproductive health clinic, offered in the same way as a pregnancy or HIV rapid test. GIFT is a social innovation. We have more than 15 years of experience in clinical and translational research to improve reproductive health and prevent HIV infection. In developing GIFT, we have the backing of the best device manufacturer in South Africa and a team of international experts in science, public health, and business, with excellent clinical infrastructure, positioning us to expand testing of GIFT into 3 regions in Africa.
With GIFT, we are innovating to improve reproductive health and prevent HIV infection in women. Public health impact is our goal.
Thank you, Lindi. And Jenna, were you going to also share some work?
Yeah, so I'll introduce Jenna now.
Thank you.
So Jenna Wilson is our next speaker, and she's an early career researcher in my group at the Burnet. She's done some phenomenal work during her honours and during her time in our group, identifying different lactobacillus species that could potentially be used as biotherapeutics or probiotics to improve reproductive health in women. So that's what she's going to be talking a bit about now. And I also just wanted to highlight that she's starting her PhD with us this year. I'm very excited about that. And she is going to be leading the preterm birth pregnancy biomarker study. So over to you, Jenna.
Thank you, Lindi. I'll just share my screen. And hopefully everyone can hear and see that one okay.
Yes, I'm just conscious of time that some people might start dropping off, Jenna, so don't take that personal.
All good. I'll just give a very brief overview. So this is the early stages of the development of a lactobacillus biotherapeutic for HIV prevention in women. So as Gilda introduced this morning, BV is linked to an increased subclinical genital inflammation and therefore increased risk of HIV acquisition. So in the optimal state of the vaginal microbiome, it's primarily lactobacillus species like Lactobacillus crispatus, which we know to be protective. They produce lactic acid, help maintain an acidic environment. You can also have a shift towards a less optimal state with more diverse bacteria that can be quite inflammatory. They can disrupt epithelia and cause this inflammation that is linked to HIV risk. So as we have the shift towards an increasing pH, we also have increasing HIV risk. The focus of my talk today really about how we need new approaches for BV treatment. So currently, if a woman has symptomatic BV, she might go to a GP and you might get metronidazole or clindamycin antibiotics. But there is really disappointing recurrence rates of over 50% of women within 12 months. And these antibiotics are, in my opinion, a bit of an outdated approach. They're broad spectrum antibiotics and they also be harming beneficial microbiota. There's always a threat of antibiotic resistance as well. So we're looking to see if is it possible to maybe replenish the optimal vaginal bacteria to reduce the recurrence of BV either after antibiotic treatment or as a separate strategy. So there are already several probiotics that are marketed for vaginal health, but none of these probiotics actually use some of these optimal vaginal strains. Some of these products use lactobacilli found not in the vagina but elsewhere. These products aren't supported by clinical evidence or randomized control trials, and they haven't been tested against HIV infectivity. These products are also usually oral capsules, whereas we'd be talking more about intravaginal products. We can just deliver these bacteria directly to where they need to go. So I did start this work in my honours, and the original aim was to assess the probiotic characteristics of these vaginal isolates. So first we need to culture and grow these lactobacilli from clinical samples, and then look at some of their properties in the lab, looking at, can they acidify culture media? Are they producing this protective lactic acid? We need to rule out and make sure they're not inflammatory, and then investigate their virucidal activity against HIV. So the samples that I'm talking about are from the MISC study cohort. These are young women in South Africa. And there's a big focus on women in South Africa, as they are especially high risk of HIV and they're a priority population. So from these 25 samples, we can then isolate different types of bacteria. So we use an anaerobic chamber. So this is very sort of sci-fi looking box we put our arms in that allows us to grow these bacteria at 37 degrees Celsius without any oxygen. And I believe there is a video produced of this that will be shared with you all as well, showing how we use this chamber. But from here, we can get these bacterial species sequenced by their gene sequencing, and then we can grow them in liquid media and assess the pH and what they're producing. So briefly, we had about 307 samples that we sent off for sequencing, and we did get 9 different lactobacillus species, including Crispardus, that nice optimal one. But I also just wanted to highlight there's so much diversity in the vagina, and I think this was one of my favorite parts of this project, was just seeing how many different species we can isolate using this chamber in these conditions. So from here, we selected 50 different isolates to further characterize in the lab. So all of these lactobacilli species did produce lactate. So this is an unprotonated form of that protective lactic acid, but there's a D and an L form, and that does differ between the species. So we'll need to keep that in mind. But overall, we did see that Crispardus had quite a low pH, produced lactic acid, which we found that lactate and of course the pH correlated. So now looking at inflammation and making sure that these bacteria aren't inflammatory, making sure they're not going to stress out human cells, we can actually grow human vaginal cells in the lab. So this is a immortalized cell line of vaginal cells, and with this we can grow bacteria at the same time. So these are lactobacilli cultures were incubated with the vaginal cells for 24 hours. We can then take the growth media liquid and we can look for evidence of an inflammatory response or cytokines. So we also included Gardnerella vaginalis in this assay. So these strains were isolated and assessed by an honest student in our lab, Sarah Hamza. And so these Gardnerella are a known inflammatory species often associated with BV. And so these were included as a positive control. So looking at the heat map on the right, all of these little colourful bars down the side, those are the different strains colour-coded by species. And then looking at the heat map, the blue is less inflammation, red is higher inflammation of these inflammatory markers. And already we can see that the blue, sort of the bottom half of the figure, seems to correlate pretty well with that yellowy Crispardus colour. And then towards the top, in the sort of dark purple and dark blue, we have those more inflammatory Gardnerellas. But interestingly, there's a Crispardus up there too. And I think what this really highlights for me is that not every strain is necessarily equal, and that's the importance of testing many different strains rather than just using laboratory reference strains. So we then wanted to see, can these lactobacilli actually inhibit HIV in the lab? So these assays were performed by Grace and David, so I didn't do these myself. But what they did was they used neutralized lactobacillus supernatant, so the liquid that these bacteria were grown in, and they actually put that directly with the HIV. So this virus was then added to T-xenombia cells. These are human cells. It's a reporter cell line, and it allows us to actually measure the relative infection of these cells. So you can see, is the lactobacillus supernatant doing something to the HIV that is maybe impacting its ability to infect cells? So we found that the most HIV inhibitory isolates were Crispardus. So in this graph, looking at relative light units of that relative infection, the lower bars are actually better. So these nice Crispardus ones down here are lower than the control, which is that dotted line across the top. But if we want to generalize and look overall at the different species, it's actually vaginalis. That was the most inhibitory species when all grouped together. So this is suggesting that the lactobacillus might be producing some sort of metabolite, maybe lactic acid, maybe other things that are impacting the ability of HIV to infect these cells in the lab. So just very brief summary. So we cultured all of these different bacteria to try and assess could they potentially be used as a probiotic to prevent BV and then other conditions in women. And in the future, we'll also need to look at things like the resistance of these strains to antibiotics. Could they be used as well as or just after antibiotic therapy? And then also send them for whole genome sequencing and look at the best performing candidates altogether. Thank you. I'd just like to acknowledge especially Grace, Sarah, and David for the assays they performed for this presentation.
Thank you, and so well done, Jenna. That was an amazing amount of work, and you've managed to keep it within our time zone. I'm sorry if I rushed you, but that was very clear. Really well done. And Lindi as well. It's such inspiring work, and I have heaps of stuff I want to bring up, but in the interest of time, we'll do that offline. But before we move on to some questions, I firstly want to thank all 4 speakers and everyone who was involved in bringing this event together. I hope you've all enjoyed the presentations and feel excited by the research being done at Burnet in this space. And we are aiming to raise $30,000 to support the research you've heard about today specifically. So if you would like to donate, you can find a link in the chat that Ash has just shared. We'll also circulate that link after with the video. And I must mention, we had an extremely generous offer by one of our donors, Margaret Stewart, who has generously pledged to match donations up to $5,000 today. So if you do wish to donate towards this research, it will be doubled. And so I really want to thank everyone for attending and their time, and it really is an inspiring body of work. So if you're able to stay on, we do have a few minutes. I'd like to open it up for any questions or general comments that people may have. Anyone? Oh, yeah, Suzanne? You're just muted. Yeah. Great.
Firstly, congratulations also to the speakers, and I'm really sorry that I wasn't here for the first half, but I was on another call. And so apologies if this question has already come up in the questions or in the presentations, but perhaps Gilda or Brianna, how does aging affect not only the microbiome, but also the potential use of lactic acid in terms of the fragility of the vaginal mucosa as women age? Did you cover that? If you did, I can speak to you later.
Yeah, thanks, Suzanne. Great to see you. And terrific question. So we've been focusing on women of reproductive age, but if you've brought a great point that the microbiome does change with age. And so when women hit menopause, the estrogen levels drop, and so does that food source, the glycogen that I spoke about. And so the microbiome does change and becomes less lactobacillus dominated. If we were going to use lactic acid, then we would probably might need to think about also combining that with a live biotherapeutic to help replenish the microbiome if there's no lactobacillus crispatus there. In terms of the fragility, that's a really great question. I would think that maybe we might have to think about estrogen as well to help, support the vaginal mucosa, because I think estrogen is used in that context. And I'm not a clinician, Suzanne, so but that's my knowledge. So yeah, look, we have been focused on women of reproductive age, but you do raise a really important comment, and certainly women who are past menopause do have sex and are at increased risk of HIV. So we need to think about that too. So thank you for your question, and I hope I've answered it in some way.
Terrific. Thanks, Gilda. And may I just follow it on? Can I have one very quick question just about, does trauma affect the inflammatory markers at all?
Yeah, I think there's
vaginal trauma.
Yeah, quite a bit of work done on trauma and trauma during sex, and I think there is elevation of markers. Maybe Lindi might know more about that too, but definitely I would say there would be increase in pro-inflammatory markers during trauma, gender-based violence, that sort of thing. There is. I know there's some individuals in the States that are focused on looking at that. Right.
Okay. Thank you. That's fine.
Yeah, if I can add to that quickly. Sorry, Jen. Yeah, so I think there's a fair amount of work on non-consensual sexual activity, but even just kind of normal healthy sexual activity, especially in the younger women, may induce some level of trauma that that might increase inflammatory markers, and there's nothing really known about that. That's actually one of the things we're investigating in an ongoing study in Cape Town. We're doing colposcopy to look for signs of injury, and then accompanying that with measuring the inflammatory biomarkers.
Thanks, Lindi.
Thanks, Lindi. And can I just finish on one submitted question for you, Lindi, that I think is hopefully an answer that's positive to end on. Can you just talk a little bit about the HIV prevention and anti-stigma campaigns that are focused on young women in South Africa and what's happening in that space that relates to your work?
Yeah, so that's a great question, and it is a very active area of research and implementation work as well in South Africa. So I collaborate with an organization called the Desmond Tutu Health Foundation, and they do the most phenomenal work in this space. They work in adolescents and young women specifically, and they're one of the NGOs that is really working very hard to implement pre-exposure prophylaxis and to increase uptake of PrEP in these young women. They've also done some amazing work, and it's not just them in isolation. There are other organizations like the Center for the AIDS Program of Research in South Africa that we also work with, and Fitz Reproductive Health Institute, and they're just like countless NGOs trying to address these issues. But yeah, there are a lot of, in addition to kind of implementing these biomedical interventions, there's a lot of work trying to increase the education levels in young men and young women as well, and trying to address stigma and improve mental health, and really and improve education around sexual health as well. And I think if you look up the Desmond Tutu Health Foundation website, you can find some really nice information about that work.
Okay.
Detail.
Wonderful. We might include that link in our follow-up. So we've come to time now, a little bit over, apologies, but we wanted to hear all about the amazing research. And I, again, want to thank everyone on the call for your support, both in being here today and supporting the Institute in general, and the vast amount of fantastic research that's done. And please, if you have any questions we didn't get to that you'd like following up on, feel free to respond to the email you'll get from Ash, and we will make sure it's followed up. But otherwise, thank you so much. Happy International Women's Day, and thank you. Bye, everyone.